Now, we reviewed gene expression, which is transcription and translation. Gene expression, as I mentioned before, involves using certain genes at certain times, and in multicellular organisms, different genes in different tissues as well. This tells you that, as I mentioned before, you're not using all of the recipes every time you make proteins. You're just going to be making the proteins that you need and not making the proteins that are not needed. This is what we refer to as regulation of gene expression. So genes can be regulated and turned on or off. High levels of gene expression can occur when a gene is induced. Induced means that it is turned on. Low or zero levels of gene expression happen when a gene is repressed. Gene repression means that we turn the gene off. This is done by different mechanisms in different cells, and we're not going to get into how, but just know that the cell regulates its genes by turning them on or off, and this changes gene expression. Your book also goes through and defines and describes the difference between chromosomes and genes, which I do think is an important thing to cover. Chromosomes are the entire contents of all of the DNA of the cell. In fact, in your cells, you have 6 billion nucleotides. 6 billion nucleotides, but only 1% of that are your genes. The other 99% of your DNA is what some scientists refer to as junk DNA. It's not genetic information, it's not recipes, it's excess nucleotide sequences. They are useful and are involved in gene regulation and other functions of the cell, but they are not containing genes. Here we have a list in Table 9.2 in your textbook showing you the genome, which is all of the nucleotides of the cell, and how many genes they contain. So Bordetella pertussis, which you can see here, this is whooping cough, contains 4 million, roughly, nucleotides, labeled here as base pairs. But the number of genes that they contain is roughly 4,000. Chlamydia, which is an obligate intracellular parasite and requires to be inside of a host cell in order to replicate, has far fewer nucleotides, only 1 million, and it has fewer than 1,000 genes. This is because it gets most of its information and most of its proteins from the host cell itself. But one of the things that you'll know that's pretty consistent is that most bacteria contain roughly 5,000 genes. The amount of nucleotides they contain is roughly 4 million. Eukaryotic cells, on the other hand, generally contain genomes in the billions of base pairs or nucleotides, and the number of genes is usually in the tens of thousands. So for humans, it's roughly 20,000. But remember, those 20,000 genes are found in only 1% of the billions of nucleotides we contain. Now, as I mentioned, the changes in DNA sequence, known as mutations, can sometimes affect the protein structure, but not always. Let's take a look at different types of mutations that can exist in cells. There are different types of mutations, the first of which is called a point mutation. A point mutation is when you substitute one nucleotide for another nucleotide. This can result in different types of mutations. The first one here listed is a nonsense mutation. And a nonsense mutation is when one nucleotide changes And that nucleotide change results in an amino acid changing to a stop codon. So for instance, if we go back to our genetic code, this would be like UGU, which normally would translate to cysteine, changing to UGA. By doing that one change, it's now changed to a stop codon. What this does is it immediately halts protein production and leads to a truncated or very shortened version of the protein. This would lead to the protein being completely non-functional. Missense mutations are a little less impactful on the protein structure. Missense mutations are when a nucleotide changes, and that nucleotide change alters one amino acid to a different amino acid. When this happens, sometimes the new amino acid is very different and can cause a change in protein function. Other times, the new amino acid, the different amino acid, isn't all that different. And if it's a similar amino acid, it will not change protein function much, if at all. Finally, there are point mutations that are called silent mutations, not listed in your table here, but silent mutations are only possible because the genetic code is redundant. This is, as I mentioned before, what you would get if you change something like UUU to UUC. It doesn't change the amino acid used, phenylalanine, and as a result, there is no change in protein function. Again, those are called silent mutations. Silent mutations would have no impact on protein function. There's also insertion and deletion mutations, and in this case, you're not changing one nucleotide for another, but instead you're adding or removing a single nucleotide. Remember that the ribosome moves down the mRNA molecule one codon at a time, or exactly 3 nucleotides at a time. So if you add or remove a single nucleotide, you're changing the codon there, as well as all of the codons down the line. This is what's referred to as a frame shift mutation because it changes the reading frame. These frame shift mutations change not just the amino acid at the location of the mutation, but they also change all of the amino acids after that. And that results in, of course, a dramatic change in the protein function. There are lots of things that can cause any of these types of mutations. Every time a cell divides, it can lead to formation of mutations. For instance, your cells with 6 billion nucleotides, you would have to copy those perfectly every time in order to get no mutations. But in fact, cells don't do that. They make mistakes. That DNA polymerase enzyme doesn't always catch its errors, and sometimes it incorporates the wrong nucleotide. And when that happens, you get a mutation. Other things that can cause mutations are environmental factors, like UV radiation, or chemicals, like asbestos, cigarette smoke, alcohol, and still others like pathogens such as HPV, the human papillomavirus, or hepatitis viruses. Finally, there are transposons that can lead to mutations as well. Transposons are not considered pathogens, but they can jump around inside your DNA, and in jumping around, they can alter the DNA of those locations, leading to mutations. Transposons are like genes that jump around, and that's why they're sometimes referred to as jumping genes. Now, these mutations are extremely important in living systems because, as many biologists will attest, mutations are the raw material for evolution. So as mutations accumulate in an organism, that causes the organism to change, and change over time is evolution. So this is essentially how organisms evolve, is they accumulate mutations. This would be an extremely slow process if organisms couldn't exchange their advantageous mutations. Because while most mutations don't do anything to an organism or have negative consequences, there are a few mutations that can help organisms. And it would be really advantageous for life on Earth if organisms could exchange their mutations and provide each other with advantages. Organisms that engage in sexual reproduction, like animals and ourselves, plants, and fungi, can exchange these advantageous mutations through sex. So essentially, sexual reproduction is all about exchanging advantageous mutations. Bacteria, though, never engage in sexual reproduction. They're simply not capable of it. This would provide them with a disadvantage, except for the fact that they have their own means to exchange DNA and exchange these valuable mutations. This is what's called genetic recombination. And genetic recombination can be done by bacteria in one of a few different ways. The first way that your book doesn't have a picture of is transformation. Transformation is when a bacterial cell takes up free-floating or naked DNA molecules from the environment and incorporates them into their own cells. These DNA molecules could come from any source. So in fact, and not to get too morbid, but when you die, you're going to have some of your DNA released into the soil. And bacteria in the soil could pick up your human DNA and try it out. Now it's human DNA for human proteins, like muscle fibers or retina, and these aren't very useful for bacteria, so they would probably get rid of them and not use them. However, if there are any cells that do find advantage with any of your DNA, then they could gain this advantage and evolve in the process. Not all cells are capable of transformation or picking up these naked fragments of DNA. Only cells that are naturally competent can pick up DNA molecules. Transformation is a process that is also used in genetic engineering and in many labs around the world. And scientists for a long time that used E. coli wanted to transform E. coli and insert or integrate foreign DNA into E. coli. E. coli, unfortunately, is not naturally competent. It took scientists over a decade to figure out how to get E. coli to become artificially competent in the lab. And it could involve either mixing E. coli with salt and putting it into a hot water bath for a brief time, or subjecting E. coli to an electrical pulse, electrocuting it briefly to disrupt the cell membrane and incorporate these naked pieces of DNA. Now that we can do this, we regularly use transformation for genetic engineering of E. coli in research labs all around the world. In the last section of this chapter, we'll talk about the other forms of genetic recombination in bacterial cells: transduction and conjugation.